Okay, this video is about quadrant streaking or isolation streaking. Going to be working with live bacteria, so we need our lab gowns, we need our gloves, and our safety glasses. Equipment we're going to need is our bacterial incinerator, so I've turned this one on and let it heat up. That takes a while. We're going to need a Sharpie or lab marker. You're going to need actual bacterial growth. This is a student sample that's been collected. We will need a fresh, clean petri dish and our inoculation loop to manipulate the bacteria. So when bacteria first grow on a plate, when we've done a sample, they've grown on all over this plate, and individual colonies start off as cells or groups of cells, but we don't know if these colonies contain one kind of bacteria, 2, 3, 5, whatever. So if we're going to identify and work with bacteria, we need to have them in isolation, so we need to have them in pure samples. And this technique is going to help us do that. So the first thing we're going to do, we're going to take our petri dish, and using the backside of the dish that has the agar in it, I'm going to take my lab marker and I'm going to mark off quadrants. So I'm just going to make a nice little X grid. That's just going to help me work. We're always going to need to put our name and the source on there as well, but we can get to that point later. Some people like to also mark the quadrants as 1, 2, 3, and 4 so that they know which quadrant is which, because when we start to work, sometimes it's hard to visualize where we've already touched. Alright, so first thing we do, we need a sterile inoculation loop. So I'm going to take my loop, put it into the hot incinerator, the tip, hold it there, and quickly I can see the tip of that wire turn orange, which means it's hot. It's now sterile. I do not then put this down. I'm going to keep holding it in my hand so it remains sterile. I'm going to hold it in my dominant hand, which is my right, and then I'm going to go to my petri dish, which contains bacteria. I'm going to find a colony that I want to work with. I'm either going to take a small sample of the colony, especially if it's a small round one. If it's a big spreading colony, I'm going to take a sample from the edge. That's where we tend to find the freshest cells. So I'm going to take a small sample. I'm going to get it on the end of my loop, and now I can close up my original plate and set it aside. I'm done with it. Now I go to my fresh plate. I go to the first quadrant, and I'm going to make a pie streak. I'm just go back and forth, a little pizza pie in one of those quadrants. I can close that, and now I need to sterilize my loop again. Sterilize my loop between each step here. Turns orange, and now I'm going to let it cool. What we're going to be doing now in that first quadrant, I have a lot of cells spread over. I'm going to take a sample of those cells and spread them into the next quadrant to start to dilute them as we go down. So by the end of the time, we'll have hopefully individual cells that are dropping off that can grow up to be full colonies. Alright, so this plate has been closed. I'm not leaving it just lying open or else it'll get contaminated. When I'm ready to go, I pick it up with my other hand. Without lifting my loop, I place the loop on the plate in the first quadrant, drag through that original streak, keep going to the second, and streak again in kind of a pie-shaped streak. Close the plate, and then I can re-sterilize my loop. It's glowing red. Pull it out. I don't want to go right back to the plate now because with this hot loop, can kill the cells that I've already put on there. So I'll let it cool again, 30 seconds perhaps. And now I have worked with the second quadrant, so I'm going to pick up my plate, use my sterile loop to go through quadrant 2, place the loop on the plate gently, drag through there, and then drag it into the third quadrant and zigzag. Put the plate down, sterilize again. Okay, now my loop is sterile once again. Now once it's cooled, I have one more quadrant to grow into. So some things that I see people doing wrong sometimes is students tend to press too hard on the plate and you'll dig furrows into the agar. That doesn't necessarily ruin your culture, but it just is a little messy. So rather than coming straight down on the plate and pushing in, kind of come in at an angle and be very gentle. You just have to barely touch. And now I'm cool. I'm going to go into that third quadrant. I'm going to place my loop on the plate, drag through that streak to pick up a few cells, and without lifting, move into the fourth quadrant and streak it on. And then I am done with my plate. I'll sterilize one more time to get rid of any bacteria that are on my inoculation loop. Now I am done with it. This plate now, I can put my name on it, and I can write the source, and now I'll put it in an incubator at 37 degrees, and it will grow out and hopefully give me some isolated colonies that I can use for staining and other tests. That's all there is to it.